So first, let me thank both Ambarish and Debashish for inviting me here. Obviously, it would have been nice to be here in person, but we'll make the best of it. So I want to talk some about transport and how it's regulated. And, you know, you've probably all seen this nice animation which shows a cargo being dragged by a single motor along a very clean, pristine microtubule. And there are, of course, many problems with this movie. One is that it's teams of motors, and we'll talk more about, you know, how we know that in a little bit. Microtubules are not clean, and there are lots of impediments to motion. And so as we start to think about how transport works, we have to start to deal with various aspects of the complexity. So, I wanted to first say, so, you know, as we think about motors and how they work, there are some obvious places where, you know, kinesin and dynein are critical. So if you think about long distance transport in neurons, for instance, where the cell body can be up to a meter away from the terminal in like a giraffe, or even in your leg, you know, you have some neurons that activate your toes and the cell bodies are in your hip. So that's, depending on how tall you are, that's a pretty far distance. And so that kind of travel distance is ineffective for diffusion. So you need active transport to drag things from one place to another. And so molecular motors are central in being able to have these kinds of structures and having neuronal cells function. And so they're also, of course, important for neurons that like to spread. So what you can see here in this little movie are individual herpes virus particles moving down a neuron. Herpes virus comes in at the terminal branches and then moves to the cell body where it replicates, and then it moves out on dendrites and spreads to other cells. So this is a way of spreading through the body without going into the blood and without being exposed to the immune system. So there are lots of neurotropic viruses that do this, and so it's of interest there too. Now, when we think about sort of a generic cell, motors are pretty important for organization of the cell and to permit a lot of things in the cell to work. They allow, obviously, the cell to crawl around. They allow the cell to bring things in and to position various components relative to each other. And we're going to be talking about, for instance, how molecular motors are used to control interactions between lipid droplets and mitochondria as an example of the kind of transport that we're interested in. But just to say that there is a lot of active transport and it's regulated. So one of the things that, you know, we start off with as a physicist is, you know, we look at these systems and we discover that they change, right? That cellular processes demand changes and regulated changes so that things happen. So for, you know, in the case of pigment granules, if you're an animal and you're green and you're on a green background, that's great. But now you're a frog and you jump onto something else. And so now maybe you need to turn gray, and so you pull all of your pigment into the center of the cell. But now maybe you jump back onto that green leaf again, and now you have to move your pigment out. And so you need to be able to, you know, aggregate or disperse your pigment in response to particular cellular sort of demands or organismal demands for cellular function. So one case where we know that there is this regulated transport is in pigment granules, where they move in or they move out depending on what the frog or the fish, you know, what color it needs to be. And so when we think about these things, we need to think about not only how do the motors work, how do they walk along microtubules, for instance, to drag cargoes to one place or another, but how is this transport regulated? So how is their activity controlled so that the cargoes go to the right place at the right time? And we also have to start thinking some about some of the challenges facing transport and how those might impede the function of the motors and maybe how the motors overcome such challenges. So we're going to be talking some about all of these things. I'll start with kind of a brief overview of some in vitro studies so that we're all on the same page about the way you go from single motor function to groups of motors and what the groups of motors do. And then we'll turn to in vivo studies and we'll talk about a couple of different case studies. We'll talk about two which are sort of more global in nature, and then the third, which is more local and involves regulation of force production. Okay, so, and I should say, by the way, that if at any point during this talk, any of you have questions, you should interrupt and ask me. I probably wouldn't, if you were to message me or whatever, I probably wouldn't see it. So either ask Ambarish to ask me or just ask directly. I certainly do not mind being interrupted. Okay, so in vitro, what have we got? So the basics obviously are, at least from what we've done in my lab, is you take an optical trap and you attach a motor to it, and then you can measure function. So if you have a single motor on a bead and you hold the bead close to a microtubule, the motor will bind to the microtubule and start to walk along. And as soon as it starts to walk, you can turn off the optical trap, allowing the motor to walk freely. And so it will drag the bead and then it will fall off and the run will end. And so doing that, you can measure the travel distance and also the velocity of the motor. And so here's an example of that for a kinesin, where you can see it goes about a micron or a micron and a half. So the bead's diameter was half a micron. So it went a couple of bead diameters and fell off. And you can say, well, in vivo, things go much further. So how might you do that? And one way you could do that would be to use more motors. And so, you can crudely put more motors on a bead and ask, well, how far does it go? And if you put a reasonable number of motors on the bead and then let it go, it'll just go and go. And so you can see this guy is going for many microns, and he will fall off when he gets to the end of the microtubule. So obviously more motors can move you further, and that's not really a great surprise, but it's nice to see that you can accomplish that in vitro. And you can also ask about forces. So how much force does a motor produce, and how do forces from groups of motors add together? So again, you take a single motor, you put it in an optical trap, and the optical trap is functions like a spring. And so basically, as the motor binds and starts to walk, the bead is pulled out of the optical trap by the motor, but the optical trap is pulling back. And so at some point, if you have a reasonably strong optical trap, the force that the motor can exert pulling you forward is balanced by the force that the optical trap exerts pulling you back, and the motor stalls. And so basically that's this flat line here that you can see right here, where nothing is happening. And at some point then, after the motor has stalled, it will fall off, and the bead will pop back into the trap center. And so by measuring the change in displacement between where the bead was stalled and the trap center, you can, if you know the spring constant, you can measure the force. And so basically the force for a single kinesin ends up being around 4 to 6 piconewtons. For most of ours, it's around 5 piconewtons. And so if you go down to a binding fraction of around 0.3, so that only 30% of your beads bind, you can get down to the single molecule level. And so when you get a bead with a motor on it, it will go up and stall at around 5 piconewtons. And if you put more motors on the bead, you'll see a lot of single motor events, but every once in a while, the bead will be in such a position or such an orientation such that 2 motors can bind. And when 2 motors bind, you see this other kind of behavior here where the force is a little higher, and you can quantify that, and it turns out that the forces are roughly additive. So for 2 motors, you get roughly 8 or 9 or 10 piconewtons. It's slightly subadditive. We won't go into the details. So that's for kinesin, and the same is true. And so that was done by Michael Vershinen, and we had I think a PNAS paper on that. And then Roop, who most of you are now familiar with. Roop Malik was a postdoc in my lab, and he studied dynein, and we looked at how multiple dyneins work together, and a single dynein had a stall force of around a piconewton, and if you put a couple on there so that they could bind simultaneously, the stall force was around 2 piconewtons, and the travel distance was much further. So motors are roughly additive in force for both kinesin and dynein. Okay. And here's a histogram showing 2 motor forces for kinesin on the left and for dynein on the right. And the kinesin study was, the dynein study, which is the top one, was done by Roop, and that was in Current Biology in 2005. The kinesin study is the one on the left here, and that was done by Michael Vershinin, and that was in PNAS in 2007. Incidentally, a little bit later on, I'll mention my website where you can download pretty much all of our papers as PDFs, should you be interested in looking at them and reading them. They're, of course, also available in the various journals. Okay, so the conclusion from our in vitro sort of side journey is that the maximal force is roughly additive for kinesin and dynein, which means that in principle, by measuring the force, you can determine the number of instantaneously engaged motors. And that's important because then in principle, by measuring forces in vivo, we can sort of determine the number of engaged motors that we're moving the cargo in vivo. So it's a way of counting motors inside of the cell, if you can measure the forces. And travel distances increase very significantly as we go to multiple motors. The exact amount for 2 or 3 depends on a variety of factors that I'm not going to go into, and I think hasn't fully been explored, but you can certainly, by having 3 or 4, active engaged motors, you can go very long distances. Okay, so we know that multiple motors can go long distances, can exert higher forces. So that brings us to in vivo studies and what's going on there. Now, when you think about in vivo studies, the challenge is that you can't grab a cargo and bring it into contact with a microtubule, because basically inside of the cell, there are microtubules in many places, and the cargoes are typically already moving along them. So you have to be a little bit more dynamic in your approach. And so what you can do is you can see a cargo that is moving, and if you can very carefully position your optical trap over the cargo, then you can measure the force. So if you position the optical trap over it and then look at how far you go before you stall, and then how far you go when you detach from the microtubule and fall back, you can measure the force. And you can do that either by having determined the spring constant of the optical trap in vivo and measuring things that way, or a sort of newer approach that we prefer is basically to measure the change in momentum of the scattered laser beam. And so then you can figure out the amount of force that the laser beam is exerted by the change in momentum. And that's described in this paper by Young-Gun Jun et al. In Biophysical Journal in 2014, which is again available on my website, should anyone be interested. So you can measure the forces, and the conclusion, so here's an example of that. So here's an optical trap measuring on a lipid droplet, so that we turned it off, and now we turn it on, we stall the lipid droplet. It tries for a couple of attempts, then we're going to turn it off, we're going to let it go again, then we're going to turn it on. And so we're making repeated stalling measurements. And when we do that, we can measure the forces. And so what we discover is that there's a peak at around 2.5 piconewtons and another one at around 5. And and this was published in 2008 in a paper in Cell with George Shubaita, who was a postdoc as the first author. So the conclusion is that, there are multiple motors. In this particular case, it looks like the single motor stalling force was 2.5 piconewtons. There are multiple motors moving the lipid droplets, and almost all the lipid droplets were moved by 5 motors or less, and 60% of them were moved by more than 1 motor. And relatively similar conclusions were found by Roop looking at a different class of cargo a little bit later when he had his own lab. So, we think in general that cargoes are typically moved by a few motors up to maybe 5 or 6, maybe a few more, but not dramatically large numbers, but not one. And so that's kind of the range of motors in vivo. Now, that number of motors is enough to move a cargo quite a long distance, at least in vitro and presumably in vivo. And so the fact that cargoes tend to move a micron or two before stopping or reversing course, we think means that there's additional active regulation to turn a group of motors on and the other ones off. And if anyone is interested later on, I'm happy to talk about that. Okay, so what does this mean now? Now that we've measured some motor function, we can see that there are multiple motors. I've said that in vitro, you can get reasonably large travel distances from multiple motors working together. You can get larger forces. We've confirmed that we have multiple motors moving in vivo. So now we have to talk about regulation, because I think the really interesting things happen when you start to ask, well, how do you change transport? How do you regulate it to achieve particular cellular goals? And there are two sort of general models for how you might control transport. One would be control of the number of motors on the cargo. So recruitment of motors to the cargo or release of motors from the cargo. And by tuning the number of motors, you could tune the transport. And that, I think, was, when I started a long time ago, that was the dominant model for how you would regulate transport. Simply, you would control it by recruiting or release of motors from the cargo. And Vaishnavi, I think, is going to talk some about recruitment of motors and how that can be used to regulate transport. Certainly it does happen. But as I started looking at cargoes, especially cargoes that move bidirectionally, reversing course, moving plus end and then minus end and then plus end and minus end, I started thinking that probably there was a lot of regulation of motor function once the motors were bound to the cargo. And we looked a lot for these bidirectionally moving cargoes. And in all the cases where we could really tell, it seemed like there was not very active regulation via, recruitment and release of motors, but rather you were changing the activity of the motors once they were bound to the cargo. And so I got interested in this sort of alternative form of regulation. And I think they both happen in different circumstances. And I think one of the interesting questions, which I'll touch on at the end, is sort of how you put these two together and when you use what. And I think we don't really know the answer to that yet, but we will certainly, I think, as a community moving forward, be talking about. So let me talk some about regulation, and we'll talk about a couple of different sort of vignettes, as it were, of how you might regulate motor function. So the first example is global regulation of kinesin family motors, and it's via this protein called CK2, which is casein kinase 2. It's a kinase, and CK2 is interesting, among other things, because its levels and potentially activity change in a variety of diseases. So it's lower in Alzheimer's disease patients, or lower activity, lower levels of CK2, I think, and it's certainly upregulated in cancer, so there's an increase in CK2. And so both in Alzheimer's and cancer, you might expect altered transport. So we started wondering about whether CK2 was somehow contributing to control of motor activity. And what we discovered is that it does seem to do interesting things to kinesin. So if you take purified kinesin with nothing around and you treat that kinesin with CK2, so you incubate the kinesin with CK2, and then you then you recruit that kinesin to a bead or a quantum dot or something, and you ask how far do the beads go, what you discover is that in the absence of CK2, suppose you had a mean travel on the order of 2.2 microns. So this is a little bit more than single motor transport. It's not, dramatically multiple motor, but it's more than single motor, because a single motor would go on the order of a micron or 800 nanometers or something. So, you have a few motors on there, and some reasonable portion of the time you're getting some contribution from a second motor to give you that added travel. So if you take the exact same amount of motor and you incubate it with CK2 and then you recruit it to beads, what you see is that the distance is more than 4.3 microns. We say more than 4.3 because basically a lot of them go, so for the purposes of this, we got the 4.3 by averaging everything that we could measure, but a lot of them went more than 8 microns. They went out of the field of view. So anything that went out of the field of view, we just scored as an 8 micron event, but it was in fact further. So that means that the mean travel was more than 4.3. So obviously there was this big increase in travel due to CK, or travel distance due to CK2 treatment. In principle, without having done anything else, you could imagine that CK2 might be altering single motor function, but we can tell you that it isn't, and you can find all the details in this Nature Communications paper with Jing Zhu as the first author, published in 2012. And so it's also, of course, consistent with more active motors. And so you can also look at this in vivo, and you can look at lipid droplets, which are moved by kinesin in the plus n direction and dynein in the minus n direction. And when you knock down CK2, either using one RNA or a different one, in both cases you see, and you confirm using Western blots that you have in fact decreased the overall amount of CK2 in the cell. What you discover is that the percentage of lipid droplets that are able to escape from the optical trap goes down a bunch. So in the untreated cells, or in those that are treated with an RNAi against a random protein unrelated to transport, we have a relatively weak optical trap, so that 85 or 90% of them, 89% versus 92% in the two cases of the lipid droplets were escaping. And then in the case where the CK2 has been decreased, now we see 23% of the lipid droplets are escaping. So a dramatic decrease in the number that are escaping, which basically means that the force production driving the motion of these lipid droplets is significantly decreased, and that's consistent with a loss of kinesin activity moving these lipid droplets. Remember that the lipid droplets are moved by more than one motor, so there isn't a dramatic decrease in the motion of individual lipid droplets. It's just the forces. You can only tell by measuring the forces, because if they had 4 motors that are active and now they have 2 motors that are active, they're still moving fine. It's just that the forces required to stall them are down. So that says that you can tune the overall number of active motors and presumably the overall number of force-producing motors on a lipid droplet by CK2. So that's sort of one mode of tuning force production. And maybe in some cases where you might be prone to getting stuck, you might alter the number of lipid droplets that are moving, but we haven't looked very carefully into that. So, and that it turns out that this CK2 mediated regulation of function is true for kinesin family members overall, not just for kinesin 1, but for EG5 and some others. And what's really interesting about this is that what's going on is that the kinesin becomes inactive over time. And so that you might naively expect that that would result from the motor just unfolding, okay? And then CK2 maybe acting as a chaperone, but that's not what happens. What happens, and we know what happens because we have a sort of fret probe, and when the motor goes inactive, the fret probe actually ends up, the two probes end up reasonably close together, and so we can tell that it's not unfolded because they'd be very far apart if it were. And so what happens is that over time, the motor goes into this stable conformational state, which is an inactive state. It doesn't bind to the microtubule, it doesn't walk, and that's apparently the active state. So presumably an inactive state, so, and that's the low energy state. So the active state is a long-lived transient intermediate, it appears, that lasts maybe an hour or so. And what's interesting about all of this is that the rate of the motor's inactivation, the rate of transitioning into this sort of stable, low energy inactive state can be controlled. And what that means is slowed by the presence of CK2 alpha. So CK2 alpha, basically, the more CK2 alpha you have around, and I should say CK2 is a complex of CK2 alpha, CK2 alpha prime, and CK2 beta. It's a tetramer. No, it's a trimer, I guess. No, I think it's a tetramer, and I think it may have 2 CK2 betas, although I'm not absolutely sure. In any event, it's a complex, and if you have the whole complex, you can actually reactivate inactive kinesins. So if you take the inactive ones, you treat them with CK2 holoenzyme, they become reactive. And so you can tune the rate of inactivation by the amount of CK2 alpha, and you can, by having the CK2 complex interact with the kinesin, you can reactivate it. And the cells actually do indeed have varying levels of CK2 alpha under different conditions. So in principle, they might do this. And if you want to read all about this, you can see a Nature Communications paper with Jingsu from 2012, and from Michelle Matson-Haas, there's another one that looks at details of the conformational change. That's a 2014 PNAS paper. Both of these are downloadable from my website, sgross.bio.uci.edu/publications, should you have interest in looking at these. Okay, so that was our first vignette. And now let me talk about our second one, which will be very short. And that's, again, a sort of semi-global regulation of kinesin family motors. And it comes about via detyrosination of microtubules to promote lipid droplet mitochondrial interactions. So when you are in a cell and you have lipid droplets and you have glucose, so, oops. Was there someone asking a question? I don't think so. Okay, anyway, if you are asking a question, just go ahead and ask. But, okay, so in a cell, oh, go ahead.
Sorry, can I ask this question? So in the
yes, go ahead.
In the case of CK2, does it involve activation or inactivation? Does any of these require energy input?
So that's a very good question, and I don't fully know the answer. Although, so CK2 is a kinase, and so it does indeed phosphorylate the kinesin. It turns out that that phosphorylation, which does require energy, which is to say, it takes the phosphate group from the ATP and transfers it from the ATP to the kinesin. That phosphorylation is irrelevant. So the activation of the kinesin by the CK2 involves a binding of the CK2 to the kinesin, and I think there's an induction of a conformational change via the binding of the CK2 to the kinesin. However, presumably that binding is somehow, since the, how do I want to say this? So what I want to say is that in order to go through the cycle where the CK2 binds to the kinesin and then unbinds, there has to be presumably some energy input, because I've just argued that the inactive state of the kinesin is the lowest energy state. And so you could imagine that you could be in a lower combined energy state when the CK2 was bound to the kinesin, but then to get them apart, you're going to have to put in energy. So somewhere in there, in the cycle, presumably there is involvement of energy, but we don't exactly understand it. I hope that answers
Okay,
your
thank
question.
you. Thank you.
Steve, this is Roop here.
Hey, Roop.
Hi, Steve. So this is the first time I'm seeing you so formally dressed, but
God forbid, yeah.
but just to kind of continue on what Debashish asked, this, inactivation, there is no role of the load from the optical trap in it, correct? It just happens even if kinesin is moving freely.
That's totally correct. However, I suspect, although we haven't fully explored it, but I suspect that the interaction of the kinesin with the microtubule may affect the rate of inactivation of the kinesin.
Okay, the reason I asked this question is we are seeing a load-induced inactivation specifically for dynein, but not so much for kinesin. We have not published it yet. So we see you have these opposing teams of many dyneins and one kinesin. Now, when the dyneins come under load, we feel that a fraction of them get inactivated, and that's how kinesin gets a chance and you see a reversal. But this is something we can discuss offline. I don't want to interrupt this.
Yeah, that's very interesting. We haven't seen any load-induced inactivation of kinesin that I'm aware of.
Correct. Yeah. So what we are seeing kind of agrees with your observations on dynein also, but let us talk about it offline because you're talking get delayed. Yeah.
Okay, great. So anyway, any other questions on CK2 before I continue? No, okay. And feel free, we can take all of this up afterwards, of course. So the second case then is, you have lipid droplets, and mitochondria in general would prefer to run on glucose, or actually the cell would prefer to make ATP using glucose rather than having the mitochondria process lipids. And so when there is both a source of lipids like oleic acid and glucose, you have a separation between the mitochondria and the lipid droplets. And you can see that in this left-hand picture where they're pretty much separate from each other. And so under these conditions, you don't really have much of a transfer of lipids from the lipid droplets to the mitochondria. The mitochondria are not using lipids from the lipid droplets. Now, when you suddenly remove the glucose so that you no longer have the ability to run on sugar, now basically the mitochondria will switch over and start using fatty acids. In order to use the fatty acids, they have to get the fatty acids from the lipid droplets. And so what happens is you move the mitochondria out on a subset of microtubules, and you move the lipid droplets onto that same subset of microtubules. And basically, by confining them onto the same sort of transport subnetwork, it's only a small, subset of the microtubules. By confining both of them onto these same microtubules and moving them along, you can promote interactions between the lipid droplets and the mitochondria, and that allows this dramatic increase of fatty acids to be transferred to the mitochondria. And so that subset of microtubules are the detyrosynated ones. Presumably, there's an increased affinity of the motors for the detyrosynated microtubules. We don't know exactly how much it is and whether it's kinesin or dynein. Presumably it's a kinesin, but we don't know. But anyway, what is clear is that by doing all of this, you can promote these interactions, and so you change the distribution of the lipid droplets in the mitochondria to promote these interactions. So that allows this transfer and increases contacts. It's discussed in this AMPK activation paper that was in Nature Communications in 2015. It's a Herms et al. And again, it's available on my website. So, so both of these examples were sort of global or semi-global. You could, of course, have a subset of the microtubules, but it's basically all across the cell. You have this set of preferred microtubules, and then you have the lipid droplets and the mitochondria moving on them and encountering each other. And of course, there's also, regulation at the single cargo level. And so I want to talk a little bit in my final vignette, as it were, about that kind of regulation. And so some of you will have seen a lot of this. I mean, it's basically published. And so I apologize for those of you who have seen it all. But I think it's a very nice example. So this is now looking at a lipid droplet in a cell, and this lipid droplet is trapped, and it's trying to escape minus end, and it fails to do so. And as it's held in the optical trap, it's going to try again, and it escaped. And now we have another lipid droplet that just moved in, and it just tried a plus end attempt, and now a minus end, and maybe another plus end. It's sort of hard to see, but there are these little blips that you'll see. And in a bit, it's going to feel very full of itself, and it will escape in the minus end direction. So we'll just be patient. And so that was a pretty robust attempt. It failed. And there it goes. Now it's out. So there were multiple, attempts, and it eventually escaped. And when we first saw this, we sort of didn't immediately realize what was going on. But it turns out that the probability of escaping from the optical trap may increase over time. And so what happens is, if you look at individual attempts, here's the first attempt at around 15 seconds, and it was a pretty short attempt, and it fell off. And the second attempt was significantly longer, and oops. And then it eventually escapes afterwards. And so we started having this idea that maybe their ability to escape from the optical trap evolves. Maybe it's not constant. And so you can quantify the probability of escape of a lipid droplet from the optical trap as a function of the escape number. And so basically escape 1 is when you first turn on the optical trap and it tries to move. That's escape 1, or that's attempt 1. Attempt 2 is anything that happens immediately after that, et cetera, et cetera. And so the probability of escape per attempt, this is not a cumulative probability. This is a single attempt probability. It changes. And so there's lots of statistics, but the conclusion was it really does change. And so that was interesting, because it looks like it's somehow adapting. It only happens in the minus end direction. So in the minus ends, you see in this case, up to about the attempt 4, there was an increase. But in the plus end, it's constant. And it, on average, if you look at everything, it increases up to attempt 5. And we don't actually stress it here, but it turns out that after attempt 5, there's sort of a reset, and you go through the cycle again. So that's sort of interesting. So there's some ability to escape, which increases, and so in the minus end direction. And so you might wonder, well, how is it achieving that, right? So we considered a couple of models. One would be a model where basically you're recruiting additional motors, and those more motors are then able to hold on longer, exert more force, and help you escape. The other, of course, is that you could change a single molecule properties of individual motors and not change the number of motors. So to sort of differentiate between these two, we looked at what was going on in vitro. So we purified lipid droplets from the cells, and we got them to move in vitro. And they were able to move quite long distances. You can see here the mean travel was, on the order of 5 or 6 microns. And what we discovered is that we saw adaptation in vitro in buffer. So we've purified these lipid droplets. They've got all of the proteins, presumably, that the lipid droplets had. And so the first attempt is, pretty short and goes up to roughly 6 piconewtons. The second one was similar, but the third attempt in this particular instance was much longer and went up to about 8 piconewtons. And similarly for the fourth attempt. So you can see that here we got 1, 2, 3, 4, roughly 5 or So attempts, and then there was a reset, and then they get longer again. And so I had told you that on average in vivo, you get roughly 5 attempts and then it resets. And that's true in vitro as well. And so the fact that we're seeing this adaptation in vitro from the purified lipid droplets was interesting and exciting, because it means that you're not recruiting motors, because when we purify the lipid droplets, we're resuspending them in buffer. There's no motors to recruit onto the lipid droplets. So we can be pretty sure that whatever is happening, it's somehow changing the function of the motors moving the lipid droplets in vitro, already bound to the cargo. And of course, purified dynein by itself does not adapt. And so that's this lower trace here where you can see we're in a multiple motor regime. So we're getting multiple motors, but you see no particular adaptation, no particular change. And the lipid droplets, the purified ones, we had minus end motion and plus end motion. We saw no adaptation to the plus end. We do see the adaptation to the minus end. So it recapitulates what we saw in vivo. So thinking about it, we thought, well, how do you change single molecule properties? Now, we already knew from our studies in vitro looking at the way dynein was affected by NudE and Lis1, and these were studies done by Michael Vershinin in conjunction with Richard Vallee's group. We knew that NudE and Lis1 changed dynein function. And so, you know, dynein is important for neuronal migration, and for that to happen, basically, for the neuron to move, actually the nucleus has to move down the axon, and so basically this is a high load event where the whole thing gets pulled, and it involves NudE and Lis1. And if you don't have NudE and Lis1, you don't, the nucleus doesn't move very well, and the neuron doesn't migrate. And that basically leads to Lis1-encephaly, which is a smooth brain. It's a non-cephalic brain because you haven't had appropriate migration of the neurons, and it's catastrophic. It's a terrible developmental disease. And that's caused by loss of this Lis1 protein, which affects dynein function. So we kind of know what happens. NudE and Lis1 are used in a high force production events. And what do they do to dynein? Well, we're able to reconstitute, that is to say, Richard Vallee was able to reconstitute, and we were able to study it in vitro, excuse me, at the single molecule level. And so if you look at dynein alone, it produces forces on the order of 1 piconewton. The duration of the force production events is a second or two, not that long. When you make a complex of dynein with NudE and Lis1, you can suddenly get these very nice long force production events. You don't change the maximum force, but you change the duration a lot. So you can last much longer. And in some cases, you can get sustained force production for tens or even hundreds of seconds. So, and you can read about that if you're interested in a Cell paper in 2010. Again, that's downloadable from my website. So the point is that we knew already that the combination of NudE and Lis1 with dynein can change dynein force production and enable it to produce longer duration events. And so that matters because when you start thinking about multiple motors and the way they work together, if when one of the motors detaches, the other one holds on, it may not be able to move forward, but if it holds on, then the second motor gets to rebind, and basically you don't fall off. And so you can get improved force production even if you're not changing the single motor force production, because by changing the detachment kinetics under load, you've changed the ability of the group to function, and that will improve the ability of the group to escape from the optical trap and overcome these serious loads. And so that's the idea behind using NudE and Lis1, is that you can very significantly change group function. And so here's an example of that. So here's a bead moved by multiple dyneins, and you get repeated attempts, but it can't really escape from the trap. But if you take exactly the same amount of dynein and you have NudE and Lis1 present, then basically you can escape from the trap because you have the sustained force production. And so, you know, in the lacking NudE and Lis1, you detach, but in the presence of dynein with NudE and Lis1, you don't detach. And so the peak forces are the same, but the sustained force production is improved. And so you can escape. So let me just, I just need to, for some reason, I end up having problems when I try and advance there. Okay, so this was actually modeled by Ambarish, who was able to recapitulate what we see. If you have a relatively high detachment rate for the dynein at the low motor level, then very few escape from the trap. And in the presence of NudE and Lis1, at that same level of force production, you get some probability of escaping from the trap on the order of 20%. Now, if you go basically to a high number of motors, you can do the same thing where maybe 40% of the beads with a high amount of dynein are escaping from the trap. And theory and experiment agree. And if you basically then put in low detachment kinetics under load due to the presence of NudE and Lis1, then you can get, you can observe this significantly improved ability to escape from the trap. And that's kind of what you just saw in the previous experiment. And this was published in Cell, and you're welcome to look at it. And of course, if you have questions, you can ask Ambarish. Okay, so this slower detachment under load allows better cooperation between motors and thus improved force production. But you have to keep in mind that in vitro with dynein with NudE and Lis1, you don't see any adaptation. You do get improved force production, and at the multiple motor level, you have better escape, but you don't see this adaptation. So the hypothesis then must be that in vivo, under normal conditions, when we're seeing this adaptation, that is resulting from increased utilization of NudE and Lis1, that somehow you weren't using much NudE and Lis1, but now after inducing adaptation, you use it more. And so we tested that. Back in the cells, we did an RNAi against Lis1 or against NudE, and we were able, you can see, we were able to get rid of the Lis1 or the NudE, and then we were able to measure what happened. And what we saw is that Lis1 is not very important for the first attempt. So the percentage of escaping lipid droplets in the first attempt wasn't dramatically different in the presence or absence of Lis1. The average peak was force was slightly lower, but not dramatically different. But what was really different was what happened over time. So over time, in the wild type, remember, you adapt, the forces go up and the persistence times go up. But in the Lis1 knockdown, the forces do not go up. They remain about constant or maybe go down even a little bit. And in the case of the Lis1 knockdown, the persistence times do not go up. In fact, they clearly go down. So loss of or lack of Lis1 is affecting this adaptation, and it's preventing it. And in the case of persistence times, the adaptation, although it's still trying to happen, presumably it's trying to switch over to using Lis1, but there is no Lis1, so things are getting worse. And the same is true for NudE. For NudE, basically, you're not getting any improvement, and you can see no improvement in force production. Now, in the absence of P150 glued, which is, central for the dynactin complex, you still adapt fine. So you don't need, for this persistence time adaptation, you don't need dynactin. We think that probably what's going on is that you're letting go of dynactin and binding to NudE and Lis1 instead, and that the impairment that you see here in the persistence times is because you're letting go of the dynactin, but you're not getting improved function from the NudE and Lis1. That's mostly speculation, although I'm happy to discuss why we think that's probably the case. But anyway, there is this adaptation that's happening that is reflecting increased utilization of NudE and Lis1. Okay, so there's no adaptation if you don't have NudE and Lis1. And that sort of NudE and Lis1 functioning in vivo and in vitro was discussed in this Nature Communications paper in 2016. Babu is the first author. He's still a postdoc in my lab and very talented. And I'm happy to talk about what he's been doing too after the talk, should anyone be interested. Okay, so how is NudE and Lis1, how is that utilization changing? How is it controlled? So we already know or knew that NudE links Lis1 to dynein, that basically NudE binds to the dynein intermediate chain and Lis1 binds to NudE, and that the Lis1 dynein interaction is in many cases transient. And there was other work that suggested that the strength of the dynein-NudE interaction could be controlled by CDK5 phosphorylation. So the model that we sort of entertained was this idea that CDK5-mediated phosphorylation was controlling NudE dynein interactions and thus Lis1 utilization. So to test that, we basically, so this is just the control experiments on the left. On the right, we did CDK5 knockdown. So we decreased the amount of CDK5. And what you can see is that we did have adaptation, but it was in the wrong direction. So forces went down, persistence times went down. So that's in the CDK5 knockdown. And there's a CDK5 dominant negative where you can overexpress an inactive form of CDK5. And the same thing was true, that basically there was adaptation, but things got worse in both cases. So both of these led us to the sort of conclusion that CDK5 really is phosphorylating NudE, and that's controlling the interaction of NudE with dynein and controlling Lis1 utilization. And I'm not going to tell you about detailed studies where we messed with the NudE phosphorylation sites and concluded the same thing. That's in the paper, should you want to read about it. The other thing that is sort of relevant here is there was this role for this 1433 epsilon protein. 1433 epsilon comes in and binds to the phosphorylated NudE and prevents a phosphatase from coming in and removing the phosphorylation. So it sort of allows the buildup of the phosphorylation. And so in the knockdown of 1433 epsilon, you don't really get this buildup of phosphorylation of the NudE, and so you don't get the adaptation, so things remain about constant. And so 1433 epsilon plays this role to help the system build up phosphorylation of NudE to allow this increased utilization of NudE and Lis1. There's another protein called KIAA0528 that also plays a role in all this that I'm not really discussing. It's discussed in this paper in 2019, which I encourage you to read about if you're interested. Okay, so in summary here, in response to load on the cargo, dynein force production is upregulated. It involves increased utilization of NudE and Lis1 controlled by CDK5. Okay, so when might it be important? Well, when you get stuck, you might need to sort of overcome being stuck. And there are these sort of bursts of active transport that were observed in the cell. And we think that was done by Steve Granick, and it was reported by Wang et al. In 2013. And so maybe that's relevant. Maybe these bursts of transport are where you've overcome some barriers. And we do see an increase in unmoving lipid droplets in the NudE or Lis1 knockdown background. So maybe they're getting stuck, but there could be other reasons for this decreased motion as well. So it's speculation. What we do know as well is that Babu was able to show that if we look at mitochondria moving in neurons, that there's drag on the mitochondria because they're basically close to the cell wall. And so that we sort of studied that theoretically with Wortman et al., and that was in Biophysical Journal. And then Babu studied this experimentally, and that was published in Traffic in 2014. And what we saw basically is that if you look at the soma, which is the process of the axon, which is narrow, there's a difference in the velocity, between the short mitochondria and the long mitochondria. The short mitochondria move faster than the long ones, presumably because the short ones have less drag on them than the long ones. And what you can do, and in the cell body, there's no difference between short and long ones. They have the same velocity, presumably because in the cell body, they're not close to the cell wall, and so they're not feeling much drag. You can use a hypotonic solution to cause the processes to expand, and that moves the cell wall away from the mitochondria, and then their velocity goes up. The long ones are still a little bit slower, but their velocity of both the short ones and the long ones is significantly increased. And we think that's because they're no longer feeling quite as much drag from the cell walls. And if you look at run lengths, the same turns out to be true. So both of these suggest that there's significant drag on the mitochondria from the cell body, from the walls of the axon, and that this may be relevant. And there are also traffic jams that occur where the mitochondria get stuck at particular places in these confined spaces. And we think that the NudE Lis1 Dynein system may be important to resolve these traffic jams and allow the mitochondria enough force production to get out of that region. And that's, again, speculation, but it seems likely. Okay, so I want to end with some of our latest stuff. I already mentioned that lipid droplets have to move to be able to contact mitochondria to transfer fat for energy production. Okay, but something that is intriguing that we recently discovered is that lipid droplets are also innate immune hubs. And so this was published in Science very recently. And what we were able to show in collaboration with Albert Pohl, who's a professor in Barcelona, is that there are all of these innate immune proteins that localize to lipid droplets, in particular in the presence of pathogens like bacteria. They get loaded up with histones, which are antibacterial and other things. And all of these proteins localize to the lipid droplets to be ready to kill bacteria. But in order to kill bacteria, the lipid droplets have to transfer these proteins to the bacteria. And so what happens is you load up the lipid droplets with these killing proteins, and then the lipid droplets trundle off and make contact with the microbes to kill them. And so here's an example where the bacteria are inside of the cells, and the lipid droplets, which are this is an EM micrograph. The lipid droplets are these black and white zebra things that are striped. And you can see they are in contact with the bacteria, and that is presumably to transfer the nasties on the lipid droplets to the bacteria to kill them. And they even, when the bacteria are inside of phagosomal membranes, the lipid droplets kind of poke through a little bit to contact the bacteria and to deliver these nasties. And we're able to show that this delivery of these things decreases the number of bacteria that are alive inside of the cells. So it seems to be a pretty effective way of killing the bacteria. So there's an interesting case where you have very local control of transport. The lipid droplets move over inside of the cell and kiss up to the bacteria. We don't have any idea how that directed transport is controlled. It may involve CDK5 and NudE and Lis1. It may involve microtubule detyrosination. This is a typo. It should be detyrosination, not acetylation. And it may be other things too. It may be recruitment of additional motors. We don't know how it is that it's being regulated, but in any event, it's controlled to promote these contacts to kill the bacteria. So I think what I'd like to end with is, this sort of general open question for the field. So, I've shown you 3 different forms of regulation. Some reflect sort of regulation of motor activity. There could also, in some cases, be regulation of the number of cargo-bound motors. There are also other ways of regulating motor activity, like post-translational modifications, which we've been studying and we've published some other papers on. And one of the things that, as a field, we don't really know is how you combine these different methods of regulation to achieve a particular cellular goal. So, which regulatory pathway is most important for a given function? Why does the cell choose one way of doing it over the others, right? And I think the answer is going to be very specific to a particular process. I don't think there's going to be a sort of general answer to this question. And I think one of the interesting things we're going to discover is that depending on what you need to do, and what your various constraints are, you're going to use combinations of these different modes of regulation. And I think that's the interesting question, is how and why do you do that? And as a field, we're going to have to figure that out. And so I'd like to pose that as a future goal for hopefully many of us here. So let me end. Hopefully I'm not too much over. The modeling was done by Ambarish. The older experiments on Dynein were done by Roop, and newer ones were done by Michael Vershinen. And then more recent in vivo measurements were done by Babu, and Dale Chapman also contributed to that. And the latest innate immune stuff was done in collaboration with Albert Pohl in Barcelona. So let me stop and take questions if people have them.
All right. Thank you very much, Steve, for a interesting talk. Now the platform is open for discussion, so we'll take the questions.
Yes, Ambarish, I have
Yeah.
a question. Steve, can I ask this question?
Yeah,
Sure.
Okay. See, you said that there is some kind of friction on the cargos in the narrow passage in axons.
Yes.
If I understand, in flagella, eukaryotic flagella, the interflagellar transport trains, when they move, they are almost, they are known to touch the ciliary membrane. So friction probably even higher there. So my question is, do you know of any similar experiment or force measurement experiments in the case of IFT, interflagellar transport?
So what I know about the IFTs is that the forces are quite high. So there has been some work that was done by what's his name at Berkeley, Yildiz, and he looked. I think he had at least one paper, and there was someone else also whose name I forget, who also looked at forces, and I think it was trains of many motors, like 20 or 30 piconewtons. They were very high forces. Yeah, so,
sorry for interruption. See, what we found that, mostly these were about reorganization by LDs at the end of, at the tip, not so much of the force generation, how many motors are involved and how they add up, etc. The reason I'm asking that we are doing some, theory of that. We have done some, it would be nice if we had some, estimates of the forces and all that, and something that we could not find. So, if you have some information somewhere, it would be nice to know. So that's sort of,
I'll look into it. I mean, I've seen force measurements somewhere where someone put a bead on the outside attached to the IFTs and measured forces on it. And I don't remember whether it was Yildiz or someone else. And it was super high forces.
No, Steve, it was somebody else. It was on this chlamydomonas flagella. We had seen that paper. I mean, I have that paper. I'll send it off, also.
Okay, great. That's the only force measurement I'm aware of.
They were huge forces, yeah.
Yeah, exactly. Maybe this is why.
Yeah, the other thing is, sort of a comment that maybe I'm wrong, but you can correct me. So I think from your talk, I guess, as a theorist, people should look at two extremes. One is, at a shorter scale, that how the, motor changes are taking place. Okay, so that this, activation, deactivation, or how the binding of, small molecules there are changing things. So that is at a sub-motor level. some theoretical modeling probably is needed to understand what changes are taking place. The other question is, who regulate the regulators? So it's probably at a larger scale that, there's a network of these regulators and how they regulate the transport. Am I right to understand that these two are that require some theories to look into both?
I think, so certainly at the very small scale, there is, we've never very satisfactorily understood this, the kinesin inactivation. We were frustrated because we didn't really understand mechanistically exactly what's going on with the kinesin becoming inactive. I mean, we have pretty good data with this, with the fret pairs saying that it's not unfolding and that it's a high ordered state, but why it's going inactive and how the hell CK2 is reactivating it is a mystery to us. So I think there's certainly room there at a sub, a very small scale. And I think there is interesting questions about who regulates the regulators. I think the first pass, though, has to be, what do individual regulators do, right? And we need to sort of take it apart. And before we start trying to build up the pathways, which I think ultimately one wants to do, we have to sort of understand, which pathways do what. And there's another really interesting thing that I skipped over, but there's, this intriguing feedback between plus N motor activity and minus N motor activity that we don't understand. This was first, I think, reported by Gelfand, who showed, that if you knock out, I think it was Gelfand, but anyway, the observation is if you knock out the plus N motors, you basically stop minus N motion too. And it has to do with, there's some sort of feedback. And so, you could imagine that if you knock out the plus N motors, everything will end up in the center because the dynein will just, drag you in. But in fact, you don't get that dramatic rearrangement, mostly because you shut down dynein too. But how that feedback happens so that it knows to shut everything down, I think is a really interesting question. And, I don't know the answer to. So I think there's a lot of different levels of regulation. And I think, our challenge as a community has to be to figure out what each one of them does, and then to work together to figure out how you combine multiple, pathways, which ones work when and why, right? I mean, I think that's really the interesting question is, given that in principle you could alter things in a couple of different ways, why do you choose this particular way? And I think the answer is going to be different in different cases. So, in some cases, you're going to have serious tug of wars. In other cases, you're going to have regulated transport where some conductor says you're on, you're off, and you avoid the tug of war. And I don't know what you do when and why, but I'm convinced there's both classes of activity. And I think that's, again, the interesting thing.
Okay, thank you.
Swamsri, you have two questions, right? Can you please ask them?
Okay, so my first question is, whenever you are, when you sort the in vitro results, so it seems that the escape of the cargo is like a rare event. So are there any first passage time calculations to quantify this? And if you compare this in vitro and in vivo results, so it may be a better way to quantitatively show that how the diffusion constants and the restoring thing acting due to the optical trap, how they're changing due to the adaptation. It is a comment and also a question.
So as far as the probability to escape from an optical trap in vitro, that's, I don't have sort of a first passage time estimate, but it's strongly a function of the strength of the optical trap. So you can tune the optical trap to be whatever you want. I mean, that is to say, you can make it so that the optical trap, the escape force is 10 piconewtons, in which case no single molecule motor is going to escape. Or you could tune it to be 1 piconewton, in which case all of them are going to escape. So the sort of probability of escape from the optical trap in vitro, in a sense, is not that meaningful at the single molecule level. It's mostly that what we do when we're looking at sort of these changes is we tune it so that the probability of escape is low, and then we do something like look at some regulatory event that increases the number of active motors, and so then we see that it increases, and it's just a reference point. And I think the same really is true for the in vivo measurements. You can tune the optical trap to be whatever strength you want, and so you can have a high probability of escape or a low probability of escape, and it's only the relative changes that matter. So we tune it so that at the start there's only a 10% chance of escaping, and that is enough to allow us to see this sort of over time, this evolution of force production, so that by the time you're done adapting, you're at the 40 or 50% escape level. But it's sort of a construct, I guess, is the point.
So I have another question. So whenever you are saying that adaptation is taking place in vivo, so I think there is some sort of learning kind of thing is happening at the motor levels. And whenever we say something is learning, certain feedback loops are involved. So I want your comment on this. So what are the feedback loops here?
So I think our answer to that is that we agree that there is a feedback loop, and the feedback loop has to be changing the activity of CDK5, this kinase, so that the CDK5 comes in and phosphorylates the NudE. And so I think that the feedback loop has to do with somehow the stress that the lipid droplet is under is affecting CDK5 activity. And so then the question is, how is CDK5 activity being altered? And I'm happy to speculate, but you should understand that this is pure speculation. So I believe that what's going on is that if you think about the lipid droplet, there is a complex of motors that attach to the lipid droplet. And there are going to be some proteins that link the lipid droplet to this complex of motors. And I suspect, and here's where the speculation comes in, I suspect that one or more of those proteins that links the lipid droplet to the motor proteins has a stress sensor in there, which is to say that there's some piece of that protein that is easily unfolded, so that when the lipid droplet is under load, which is to say, the lipid droplet is being held and the motors are trying to walk, and so there's tension that is built up between the motors and the lipid droplet. I suspect that what happens is that there's an unfolding event of a protein in that linker region between the lipid droplets and the motor proteins, and that folding event then basically recruits the CDK5 from somewhere else on the lipid droplet. Maybe it's floating around on a tether and it's not particularly close to the NudE. And when this unfolding event happens, now the CDK5 is held in place close to the NudE and is able to phosphorylate it. And so the more you have unfolded this protein, the more the NudE ends up being phosphorylated, because the more the CDK5 binds to this unfolded piece of protein. That's my guess. But and that would be sort of the feedback loop. but I don't have any evidence for it. I don't know what the protein is, and I have no evidence for sort of direct recruitment of CDK5 to a particular location. So it's all speculation, but that's what I think is going on.
Thank you.
All right. Any other question? Then, okay. Let me also ask 2 questions from my side. One of them is an easy one. The other one might be slightly difficult to answer. So the first one is that I looked at the pictures where these lipid droplets are going and attaching to the bacteria, and there are stripes, right, on these lipid droplets. What are these?
Oh, so the stripes come from an artifact of the electron microscopy. So basically, depending on how you do the fixing and staining, you can either have the lipid droplets be white or black, or you sometimes see these stripes. I'm not enough of an EM person to know what you get when, but that's what's going on. It's just an artifact of the EM processing.
Okay, there is a question from Amir. Amir, can you please go ahead and ask the question? Then I will ask my question later.
Hello.
Yeah.
Yes.
I was asking. You have already answered in Sam Shari's question, but the question was like, what are the order of forces you are applying in optical trap? Like, if one uses a strong optical trap, maybe the attempts to escape of the protein will be unsuccessful. Like, so I was asking.
So typically, our optical trap has an escape force on the order of 10 to 15 piconewtons. We can tune it up to about 15 or 20. You start to get, in ViVO, you start to get optical damage when you go up too high in power. So that kind of limits it. In vitro, you can sort of mess some with the size of the bead and you can tune things. But generally, you know, when you see these sort of lipid droplets escaping from the optical trap in ViVO, probably the escape force is on the order of 8 or 10 piconewtons, something like that. I'm not sure in those particular experiments what it was, but it's not more than 10 piconewtons, would be my guess.
Thank you.
Okay, Farnoosh, you had a question. Maybe Steve can quickly reply to that. Farnoosh?
Yes.
Yeah.
Hi.
Can you please ask your question? Yeah.
Okay, I want just know about the process in which you extract motor molecule.
You mean, how do we purify them?
Yes.
So for the original studies of Dynein, they were basically purified by our collaborator, Richard Valley. And I think Roop's lab has actually more direct experience purifying Dynein than we do. So whatever he tells you would be more relevant than what I can tell you. For the kinesins that we study, we mostly make them. We express them in E. coli and we have a tag and we purify them. I'm happy to provide you with a protocol if you're interested for that. For the lipid droplets, where we're studying the intact lipid droplets, it's a pretty simple purification procedure where you feed the cells a reasonable amount of fat so that they make lots of lipid droplets. And then you break apart the cells using a Downs homogenizer, and then you float the lipid droplets through a sucrose gradient. And so the lipid droplet, you basically spin them using an ultracentrifuge. The lipid droplets float, and you pull off the lipid droplets from the top of the gradient, and those are your active lipid droplets that that move along the microtubule. So that's how we purify them.
Thank you.
All right. So Steve, my second question. So you talked about the motor regulation. I mean, there are different ways in which motor activity can be regulated, and one of them is basically by altering the track, right? So one of the possibilities you mentioned was post-translational modification. The other one, which is also, I mean, there is also some evidence from some of your work that the microtubules from cell to cell have certain variations. And this is typically due to the variation of isotypes. So what do you think about it? Is it hard coded in the cells, or there is something which soft coded and changes as the cell with the cell life?
So I don't know about all post-translational modifications. Certainly, the case where we understand it the best is this case where it's the detyrosination of the microtubules. And so in that case, the detyrosination is being controlled by AMP-K. And it's not, I mean, it's a regulatory cascade which ends up detyrosinating a subset of microtubules. I don't know how you select which ones are the ones that you detyrosinate, but it's only a small subset of them, and it's a regulated thing. So you can have dramatically different amounts of detyrosinated microtubules depending on whether, you know, glucose is present or absent. So it changes dynamically. So I think in general, it's going to be dynamic and it's going to be changing. You know, there's also cases where things like acetylation seem to change based on how old the microtubule is, right? And so there's probably those kinds of regulatory events too. You know, the thing that has been, I think, a little hard is that the magnitude of effects one sees for the affinity of the motor to the microtubule isn't that dramatically different depending on these different post-translational modifications. And so it's hard to know how that post-translational modification ultimately ends up exerting its effect. So like in the case of the detyrosination, it was clear that that's what it was, because we can mess with detyrosination and we can mess with the whole process, the whole, we can, you know, by altering detyrosination, we can alter the number of interacting mitochondria with lipid droplets. So it's clear that it's regulated by that, but we don't exactly know how much affinity change there is for which group of motors in the detyrosinated versus the normal microtubule. So it's a little hard to know how to, you know, play it out. So I think that's the thing. But I think in many cases, there are clearly subsets of microtubules that are identified via some process and that, you know, didn't exist before in a cell, and then they do exist after something happens. So I think it's a pretty dynamic process. What I don't know about that is whether or not such a dynamic process can be used to control delivery to a specific part of the cell, right? Does it happen globally when you sort of change detyrosination so that everything goes out? Or do you sometimes have it where you locally alter functions so that you get delivery to a particular place? You might or might not, I don't know. I think that's an interesting question.
Any other question from anyone?
I'll just make this more of this group here, Steve. This is not a
Yes.
question, but rather a comment. In this, your recent paper where you show with Albert Paul that about these lipid droplets are immune hubs, I really enjoyed reading that. One very interesting point is that in one condition where you have lipopolysaccharide or don't, I kind of forget, that in between the lipid droplets and the mitochondria, you have extensive invagination of the ER membrane.
Yep.
Okay, and that's really exciting because, you know, my feeling is from some work that you have been doing is that these lipid droplets actually, most of the time, they're tracking the endoplasmic reticulum membrane. Okay, they are moving along that, and we have separate evidence for that. And And so this somehow the mitochondria, the ER and the lipid droplet, their localization possibly along microtubules is being controlled in a way that we don't quite understand. And the reason I say this is that we have now been able to reconstitute lipid droplet ER interactions in vitro. Okay, so we basically deposit an ER membrane on a cover slip, and I'll write to Albert also, and we want to do this in cells treated from lipopolysaccharide or not. Okay, and see if the interactions change. And if we can do it in vitro, we can figure out what's going on there. So just a comment.
So I think that's a very interesting thing to look at. And I think, one of the things that, the thing that, hopefully came through in this paper that I was so excited about was the fact that, and we didn't make a huge point of it, but I thought it was really cool. So, these antimicrobial proteins like histones and cathelicidin that kill bacteria, they will do very nasty things to mitochondria because, of course, mitochondria are pretty close to bacteria, right?
Yes.
And so what's really cool is you see, as you load up the lipid droplets with these nasties to kill bacteria, you see this dramatic separation of the mitochondria from the lipid droplets, presumably to protect the mitochondria. And so, and that part of the interaction we understand, that's, via, it's talked about in the paper, right? But you're right that there is this other part of things where they're interacting with ER, and that we haven't really studied or thought about that much, but it's clearly going to be dynamic and presumably going to involve RABs and other things. So I think that's a very interesting question.
Right. Yeah. Thanks, Steve. Yeah.
Any other quick question or comment from anyone else? Okay, if there are no more questions, then I would like to thank Steve on behalf of everyone present here for a very interesting talk and very interesting discussion thereafter. All right. Thank you very much.
Thank you.
Yeah.
I enjoyed it.
Yeah.